Hello students, in this video lecture we are going to discuss another interesting topic from the chapter of the sexual reproduction in flowering plants, and that too the pollen grains. Alright, so we have already talked about the anther structure, and we have already talked about and discussed about the microsporangium and the process of microsporogenesis that results into the process or formation of the pollen grains. Now in this video lecture, as you can see on your screen, the model or the cross section of the pollen grain being represented over here. All right, now so basically in this lecture we will be observing that what are the different labelings or what are the different components of a pollen grain, what are the special features that makes pollen grain to represent as the male gametophyte, and what is its importance. Alright, so let's start with some of its basic features first, and then we will go on with its labeling part. Alright, now so when we talk about pollen grain, then pollen grain is considered to represent the male gametophyte. So you can see the shape of the pollen grain is usually considered to be as spherical. Alright, so the shape is spherical. And what about the size if we consider? So the normal size of pollen grain is considered to be around 25 to 50 micrometer in diameter. Pollen grains, they, if we consider the external structure of pollen grain, we will be discussing the pollen grain in two parts. The first part we will be considering its wall, and the second part we will be considering its internal structure or component. Alright, so if we consider the wall of the pollen grain, then wall of pollen grain is basically of two layers. So wall of pollen grain is two layered. Now in these two layers, the first layer, that means the outer layer, is considered to be as exine, and the inner layer is considered to be as intine. So we will also be seeing in the model as well that where these exactly the layers present. Alright, now the exine is basically the outer layer, and this is also thick and hard. So this is the outer layer of pollen grain, which is thick and hard also. Now the main important point of this outer layer is that this exine layer is usually made of Sporopulenin. So this is made of a very resistant chemical substance which is called Sporopulenin. So Sporopulenin is actually the chemical constituent that makes up the exine layer of pollen grain. Alright, now so let me just make you people see where the exine layer is being present. So as you can see in the model, let me just flip this model a little bit. So you can see the outer hard crusty nature of the model or the pollen grain. Alright, so if I just isolate, that means see this part, if I just label it, then this part we can consider it as to be the exine part, which you can see that it is usually exhibiting the crusty nature, or you can say fascinating patterns and designs. Alright, and if I just isolate this part, if I have to isolate only this part, so you can see this is usually made of crusty nature. Alright, that's why this is considered to be as the exine layer. Alright, and this is usually exhibiting the fascinating patterns and designs, which is actually not found in case of the intine layer. And when we consider this intine layer is basically the inner layer, which is thin. Alright, it is the inner layer of the pollen grain. So if I just have to label this part, then we can just flip this model a little bit like this, and then this layer, which you can see, this one is representing the intine. which is just next to the plasma membrane. Alright, so this one next to the intine layer is basically the plasma membrane. So if we see the continuation, then what is the sequence? So the first layer is the exine layer, which is the outer tough layer, thick and hard, made of sporopulenin, and the next layer is the intine layer, which is thin. Alright, and the composition of this intine layer is that this is made of cellulose and pectin. So this is about the wall of the pollen grain, which is 2-layer. Now, let us study a little bit of detailings of the sporopulenin, because exine is one of the important layers of the pollen grain, and the presence of the sporopulenin makes that layer to be very important. Alright, now let us just talk about only the sporopulenin part. Now, so when we talk about sporopulenin, then sporopulenin, we know that this is the outermost hard covering of the pollen grain. Now, sporopulenin is considered to be as one of the resistant organic materials known. And it is so much resistant and it is so much tough that it is going to resist the damages coming from the high temperature or acids or alkalis or any other enzyme. So it basically can resist damage from high temperature, acids, alkalis, and other enzymes. This is very surprising to know that none of the enzymes have been found that can actually degrade sporopulenin. Alright, and because of this property of resistance, pollen grains are well preserved as fossils. Alright, so due to sporopulenin, pollen grains are well preserved as fossils. And this question has been asked in the examination a couple of times as well, that why pollen grains are preserved as fossils. So this is because of the presence of this sporopulenin in the exine layer. Alright, now the next thing that we are going to look into are the germ pores. Alright, so as you can see that there are some places in the exine layer where the sporopulenin actually seems to be absent. If I just make you people see in the model, so you can see that these are the points which you can see. Alright, these are the points where the sporopulenin is supposed to be absent. Alright, because sporopulenin are present over here only, this part only. They are covering only this part. Alright, these parts, while the parts which I have circled, that part is devoid of sporopulenin. So that part ultimately represents the germ pores. Alright, so we will now discuss in detail about germ pores. These are, these points are considered to be as germ pore. Like here also germ pore. So germ pore are basically the prominent apertures. These germ pore, these are prominent apertures in the exine layer only, where you can see in the figure itself that the sporopulenin is absent. Now the reason of absence of sporopulenin in these sites of germ pore is that the function of the germ pore is to provide the space for the emergence of the pollen tube. Alright, if we just specifically look into the function of the germ pore, alright, then the function of germ pore is usually to provide the space for emerging out of the pollen tube. If this sporopulenin would have been continuous, alright, so if this sporopulenin would have been continuous, so we know that this sporopulenin is very much resistant, so it won't allow proper amount of space for emergence of the pollen tube from the generative cell. So that's why the Sporopulenin is actually absent from those sites where the germ pores are present. So the reason is because of that only, in order to provide space for the emergence of the pollen tube. Alright, so function is to provide space for emergence of pollen tube. Alright, so this was about the germ pore part. Now, this was all about the external features that we have seen in the model. Now, what about the internal structures that we are seeing? We will now talk about that part. Alright, as you can see that initially the pollen grain is considered to be as one cell. But when the pollen grain gets matured, the one cell divides to give rise to two cell. So basically the mature pollen grain turns into a two cell stage. So now what we can see here in the model is the two cell stage of the pollen grain. So here we are looking at the two cell stage. This is the two celled stage of pollen grain. So we are looking at this part. Alright, now what are the two cells? So the two cells are this bigger cell, which you can see is being represented as the vegetative cell, and this one, the smaller cell, is considered to be as the generative cell. So these are the two cell stage of pollen grain. Alright, so this is the case. Now this vegetative cell, as you can see from the model itself, that this is usually large in size. And it is usually generally spherical in shape. And the function of this vegetative cell is that since it is large in size, so it will be having abundant amount of reserve food material. Alright, so it has abundant food reserves. Now, when we see the generative cell, alright, so the generative cell is usually smaller in size. So this is small in size. And you can see that this generative cell is actually floating in the cytoplasm of the vegetative cell. Alright, so that is the case. And this is usually spindle shaped. Here the reserve food material is nil. So the function of the generative cell is to divide mitotically and to produce the two male gamete. So the function of generative cell is to divide mitotically to produce two male gametes. Alright, so this is the function of the generative cell. Alright, so we have seen the internal structure, that the two cell stage of pollen grain, once the pollen grain gets matured, the one cell pollen grain divides to form a two cell stage, and the two cell stages are the vegetative cell stage and the generative cell. Alright, so we will see further labelings in the cytoplasm of the entire this vegetative and generative cell. So this one which you can see is representing the green part, which you can see is representing the vacuole part. Alright, and this one is considered to be, the central part is considered to be as the vegetative nucleus. So this is basically the vegetative nucleus. And this one is representing the generative nucleus. So we have seen the internal structures as well, about the external structures as well of the pollen grain. Now we will see some of the little informations about pollen grain as well. The shedding of the pollen grains, that means the release of the pollen grains in case of the 60% of the angiosperms after being mature, you can say. Alright, so in case of more than 60% of the angiosperms, the shedding of the pollen grain usually takes place at two cell stage. Alright, that means the two cell stage which I have mentioned it right now, which is vegetative cell and generative cell. But in case of the remaining of the species of the angiosperms, it is at three cell stage. So this is also a very important point that has to be noted, that if we consider the shedding of pollen grains, there are two conditions, that in case of more than 60% of angiosperms, the shedding takes place at 2-cell stage. The entire description of the content is already being provided in the description section of the video link. Alright, so you can go through it. So this takes place at 2-cell stage. 2-cell stage, we have already seen that it is vegetative cell along with the generative cell. But in remaining species of the angiosperms, if we have to consider, the shedding usually takes place at 3-cell stage. Now, how come the 3-cell stage is formed? The 3-cell stage is basically one cell is that of the vegetative cell, and we have already seen that the generative cell is going to divide to give rise to 2 male gamete, along with the one vegetative cell makes up the 3-cell stage. So this is 2 male gametes from generative cell. So this will make up the 3-cell stage. So this is very important and that has to be remembered. Alright, now apart from that, pollen grains, since pollen grains, they are very minute in size. So pollen grains are also responsible for causing severe allergies in case of humans. So, pollen grains also causes allergies. And that allergies ultimately leads to various kinds of diseases like asthma, bronchitis, etc. in case of the humans. Alright, now the pollen allergy for the very first time was caused by a very predominant weed which is called Parthenium. So, pollen allergy caused by Parthenium, which is also known as carrot grass. Now this Parthenium, it has came into India as a contaminant with the imported wheat from the USA. Now apart from that, pollen grains, they are also considered to be rich in nutrients and are basically used as food supplements in the form of food tablet. So pollen grains are specifically rich in vitamin B12. And they are very much rich in minerals and amino acids. And since they are so much rich in nutrients, that's why they have been predominantly used by, and as a food supplement, they have been predominantly being used by athletes and race horses in order to increase their performance. Alright, this was some of the important features. Now apart from that, another speciality about pollen grain is that, pollen grains are also stored. And they are stored at a temperature of minus 196 degrees Celsius. That means in liquid nitrogen. So if we have to, if for further studies, you can say, or for further crop breeding programs, if there is any breeding programs that has to be in which the pollen grains has to be used of a particular plant species, then these pollen grains are stored, and stored pollen grains can be ultimately used. So storage of pollen grains is basically at minus 196 degrees Celsius in liquid nitrogen, and that too, it is specifically for crop breeding programs. So this was all about the informations of the pollen grain that we have seen. So we have seen its structure, internal, external, we have seen its advantages also, and we have seen its disadvantages also. Alright, thank you.